Okay, so I'm going to talk about not so much on the background of things, but on what we built, motivated by what is happening because of all this AI revolution in structural biology. But also I give a few examples in a few projects, which might seem very naive to a lot of you, and I'm very well aware that many people in the audience have done much more complex things with AI, but I'll give it a go anyway. So I'll be a bit emotional in the beginning, because you know, when I first seen what happened with Cascais, you know, I thought it's one of these publishers' scams, but then it's Cascais. It's like, so the next stage, of course, it's like insane. And I went on bargaining, you know, like why, you know, I heard the rumor that I'm retiring. Who told me? Pedro told me that I'm retiring. Perhaps it started from that slide. You know, and you can't, I'm not going to get a grant for that again. And then the acceptance stage came in a strange way, because Andrew asked me and my close colleague Tizia Sixma to write a review article. And we indeed wrote that article, which you can get the QR code. And I think also that it's a couple of years old by now. I think it captures the essence of what is happening because of AI. And the remaining feeling, quite seriously now, is excitement. It is the most amazing thing that I think has happened in structural biology. It's free servers, free code, different initiatives. We always talk about AlphaFill, but you know, the Rosetta projects and David Baker's team, you know, like they've done a more than amazing job that I'm sure it will be recognized quite widely and very soon. But let's not forget that, and let's not forget the effort that the ABI has put on this AlphaFill database, that if anything else, it's saving a lot of CO2 emissions by rerunning the same jobs over and over again. And recently, of course, you've all seen the Nature advertorial about AlphaFill 3. It was presented as a research article. And it's a pretty good article, I have to say, but it's really annoying. Like, you know, if you or me would publish in Nature or something new these days without the code and without making available executables to the community, and with putting limitations not only on the number of jobs that you're running, but also on the scope of the jobs you're running. Read carefully. You cannot do screening, according to the disclaimer. You cannot, of course, do anything that could be of commercial value with AlphaFill server. And, you know, like, okay, it's their software, but I felt a bit betrayed by our friends in the publishing world. It's not the first time I feel betrayed by Nature, by the way. So, the first thing that I had looked at AlphaFill back on the day that was there, it's this very weird project. So it's a very long-going project. Since then, my institute has not only lost Pete Borst as a director, only as a director. Pete Borst is alive and kicking in his 90s, and he's still interested in the project. We're on the 4th director by now. But this protein is binding a glucosylated thymidine in the DNA of protozoans, of kinetoplastic protozoas. Very importantly, Leishmania and trypanosomes that you know from the Onimus diseases. So when actually a similar protein was a target in Cas14, it was predicted perfectly by AlphaFill 3, so I got to see that early on. It was very impressive. It could even be used for molecular replacement. And you know, like, so what we've done in that protein when AlphaFill was coming out, we realized that this domain for which we have solved the structure was right in the middle of the protein, right here, this blue. And we realized if we splice it out, which seems like a silly experiment, what we would get a protein made from the N-terminal half and the C-terminal half, which was not only soluble, but it was perfectly active in its enzymatic function. Which was, we did some SACs, we found out this blue domain is very likely mobile and the green domain is very solid. We did some smart gel filtration experiment with 3C proteases, if you get it, like adding a cleavage site. So the N-terminal and the C-terminal half, we determined experimentally that they were staying together and somehow that domain has inserted itself between us. So that was the picture we had when AlphaFill came out, that you have a thymidine hydroxylase made by the N and the C terminus, and the DBD, the DNA binding domain, is inserted in the middle. So you can imagine how stunned I was when I've seen this 100 kilodalton structure predicted perfectly well by AlphaFill. Because this is here the N terminus, this is the C terminus. You see a shared, so you have 4 strands of that and another 6 strands of the C terminus, making a continuous beta sheet. And AlphaFill predicted this very stable interface between the dark green and the light green, the N and the C terminus, perfectly well. It also predicted that this blue domain, the structure was perfect, of course, as well. But what I found really intriguing is that you see in these infamous PAE plots that was, you must look at them. Don't look at the structure, look at the PAE plots. That's what I say to everyone. So you see, it tells you that the position of the N and the C terminal domain is relatively well known. So there's no error in the position of this residue relative to that position. But although this fold is known very well, it could not guess its position in relationship either with the N or the C terminal domain, which is in perfect agreement with what we already knew experimentally from biochemical and SACs experiments. So now the stunning thing is that look at this little line here, or there, if you prefer. What is this telling me? It's telling me that the position of the N terminus and the position of the DNA binding domain are very likely correlated. And although I was working on that protein for 15 years, I never felt that. We had no experimental indication for that. So what we did is like, okay, like, is that true? Like that this is the N terminus, by the way, in gray, so not only it's close, but it really claimed that the relative positions are well known. So we said like, okay, let's go ahead. And we looked at an old experiment. And this is the DNA binding affinity in orange for JPP1 protein, and in blue of the JDBD domain alone. And one is around 14 nanomolar, the other one is 19 nanomolar, which I wrote on the paper. They're virtually identical. Why virtually? Because the standard error was 0.3, but you know, like 17 nanomolar and 20 nanomolar, who cares, right? But if you put them next to each other, you know, and you put the log scale, you do see the difference. So what we did then is that we deleted the N terminus from the JPP1 protein, and if we delete 23 residues, you see now the magenta and the blue line are overlapping perfectly. So what does this tell me? It tells me that the C terminus is contributing to the affinity of the full-length protein. If we get rid of a bit more of all this gray area, we lose a bit of affinity, which, you know, we overdid it, we destabilized the protein very likely. So we could get, like after 15 years worth on that protein, literally, like new insight, just by looking carefully at the AlphaFill prediction, which might tell you that, you know, we're a bit silly, we missed it, but I don't think so. And this is the model that we have now, like that we have the blue domain that binds DNA, we have the green domain that binds DNA, and we knew that this distance from genetic experiments should be exactly 13 base pairs, and the N terminus we thought it's something like a safety belt. And keep that in mind for the end. I will switch gears. Anyway, that was published. So the other thing that we missed there is like JBP1, I didn't tell you, is an iron-dependent thymidine hydroxylase. There should be an iron here, and there was no iron. And that's what gave rise to the AlphaFill project, because we started noticing very early on that all, this here is myoglobin, it doesn't have its ligand. Hemoglobin would not have its ligand. Hemoglobin would not be a tetramer. And actually, this is there to also remind you that in the popular media, some scientists say that AlphaFill solved the protein folding problem. It did not solve the protein folding problem. It did not provide us any insight to protein folding. All this AI is amazing in predicting structures. That's what it's doing. It's predicting structures, and these proteins should never be folded. Myoglobin does not fold without heme and iron, but its structure is predicted perfectly well. So since we predicted so well, we set up and we said like, okay, let's find all the cofactors of proteins and common substrates and everything, and let's put everything together, and that's how we created the AlphaFill data bank. That basically here you see, for example, myoglobin, and now myoglobin gets all the possible ligands that it has. We made like 12 million transplants on the ABI database. By now, when we started with 1 million models. There's another million things run by users by now, and all this in this data bank. That's how it looks like. That's it's the retinoic acid binding protein. You see the substrate, you see all the possible substrates. You can click on what you want to see, you can zoom in, and you can even energy minimize it. It's published, the server is there, play with it. And of course I looked back on JBP1 and there was no iron. Why? Because JBP1, there is no homolog in the database, and we need sequencing identity of at least 25% for anything to work. AlphaFill is not that smart. It's basically a structured superposition algorithm that is very well engineered and with some very nice tricks and validation, of course, but we need homologs. JBP1, no homologs, no iron. So we said, like, okay, let's try to see if we can build an AI that would recognize cofactors that bind to proteins without any sequence or structural similarity. And let's start from an easy case, like metals. So the way that we thought of that is that most AIs, they think of proteins like graphs, right? It used to be connected alpha carbons, connected residues. Now the new AlphaFill 3 and Rosetta all atoms is connected every single atom, and so to make relationships. But like proteins, if you see our space-filling models, they're solids, there are soft solids. It's a bit like, they have their clefts, and you can push it out a bit, you can twist them a bit, so beautiful examples of how they move. There's a bit of, it's not like shaped pillows that you can bend and move and so on, but you cannot put something on the inside. So we thought, okay, let's treat them as 3D solids, and let's treat them as 3D pictures. Pictures like 2D pictures are made from pixels, 3D pictures from voxels, and in pictures you have colors. So we made up some colors of atoms that have different properties, like that's what we chose. And that's, for example, how here asparagine would look like. Gray would be for the carbons, red would be the oxidants, blue the nitrogens, in a simple example. And once you code that in the computer, you take your protein, you put it in a box, and you make a 3D picture of it with the voxels being colored by property. So now you do some little tricks. You make what you call the sampling cube to sample every single metal binding protein of specific metals. Then you move the cube around, and then actually in every move you have what is called the learning cube, which is the bit that you rotate around. These are smart tricks, but they're out of the book. None of them are ours, and definitely Unit++ is not ours. It's a very standard way to learn about pictures, and that's what we did. We described the pictures, we redimensioned a few things that was actually going from 2D to 3D was fun. And then actually we learned how zincs, calciums, magnesiums, coppers, and irons would look like in proteins. So once we learned it, we can take any protein, present it as voxels, and get this sampling cube running all over the protein and see if we could find any of these metal ions. And that would be the answer. If you know, like here, it found probabilities for magnesium, for copper, maybe for zinc. And of course, it finds lots of things on the chemical environment. That's how it's not a single zinc. It finds lots of voxels that could be attributable to zinc, because zinc has a volume, remember. And then we filter these, we choose one, we do some standard tricks, and we come up with metal ions. And it works pretty well, like, you know, like there's a zinc in the polymerase, we find it in the reductase, we find it in iron, calcium, we find it, even two coppers that were next to each other, we would find them very well. We could also get funny things, like, you know, we found this magnesium coordinated like these waters. That's not there in the PDB structure, but, you know, like we find it. We miss a few things, we don't get this copper in that protein, which is, you know, very likely we miss it. And this is like another fun example that we find zinc while the protein has nickel. And of course, like you wonder, does the protein really have zinc or nickel? I would bet you this is a zinc binding protein, and the nickel comes from the purification. Very good recall rates, well above 90%. These are some other cases. Actually, zinc fingers are lots of fun because they are really well ordered by AlphaFill, but the zinc is missing, and a few other things we recovered. So the next thing, of course, was like, can we only do metals? No. If you did other things that are solid with no rotational symmetry, rotational freedom, you find other things. So what you find here is that we taught it to look for phosphate groups, diphosphate or triphosphate, for riboses, and for nucleotides. Actually, we're a bit lazy here, only adenine. And then, like, if three of them are predicted to each other, what could it be? It could be ATP. And actually, these are all from the test set. We did not learn on this, we left them out, and we can recover the three moieties, and then we can fit, using some other algorithms, ADP or ATP, minimize it. We don't have a brilliant discovery rate, it's just about 50%. This is a threshold issue actually, which Ren is working on. I think already that this will be above 80%. We have, so this will go down. We find quite a few things that, again, like by a manual check, this is very recent results, by a manual check you start realizing that these are very likely proteins that should bind ATP, but they don't have ATP in the PDB. There was this paper a few months ago, the dark matter of metagenomes, that they found new faults, that they could not find structural homologs or sequence homologs to these. These are hundreds of thousands of proteins, actually millions to be exact, and they defined 13,000 new families. Each family has many members. We took a representative member of this, we looked for nucleotides, we found 303 likely nucleotide binding domains that could not be detected by sequence or by structure similarity. And actually, we made 4 of them, we produced them, we tested them, and at least since yesterday, the 3 that we could that we measured, they were all binding DNA. So experimentally, what we find, even on totally unrelated new faults from weird bacteria, from weird metagenomes, is more or less true. I will go very briefly on that because Jose Maria might remember that. This is cryo-EM done in his lab. And although we published it quite happily, we could never understand what it really shows us. It was too thick to be a coil-coil, too thin to be a hexamer, too short to be that, and so on. We had the crystal structure of the N-terminus, it was a coil, it didn't help. We did a lot of sacs as well, and again, the sacs, it was not a long coil, the length was wrong. If you would fold in the middle, it would not work. It was a mess. We had some also mass spec data that followed that as soon as we run Spindly as a dimer in the AlphaFill 2 server, it is this very weird thing that we knew that this dent at 160, that it would fall back to itself, but we have failed to predict the possibility that also around 360 another coil would be formed. And when you take all these and you map this to the mass spec cross-linking, it makes sense. And me and Andrea Mosacchio, we were doing this independently. When we had all the experimental data, we could not make sense. We were even arguing if it's a monomer or a dimer or a trimer. As soon as AlphaFill was out, we picked up the phone and said, we know how it falls. Yes, we know. Let's write a paper. Good. Another one that we did, that was a project that Patrick was doing, who's also in the audience, is there was this huge protein timing that our colleague Arnold Sonnenberg at the NTI works on, and it's very modular, different domains. He came to us and said this strange R7-R8 module is binding the C-terminal helix of Cascais. So it was really binding. We measured the affinity, made the crystal, made the protein, found molecular replacement solution because of this angle and the symmetry between the domains. We had the structure. And then Arnold goes and he does the experiment, which is get rid of R7, R8, and Cascais was still binding. By the way, little control experiment, AlphaFill, you see, it tells you it doesn't bind to 7, this is very red, but it binds to 8, which is very blue, and this is indeed what is happening. So what you do is that you go and you run a few jobs, copy, paste, click, enter. I admit I did it like that. And then you find out that, for example, R11 is very blue. R8 is very blue, I already told you that. And actually, R3 is very blue as well. So what you do again, you go in the lab, you make all these constructs, and you see R1, the ones that are important, they're still binding. So the R3 that is binding, when you delete R1, R3, you lose binding and so on. So all these were experimentally confirmed as well. Another very recent project that we released in bioRxiv only last week, it's the genome organization, how genome is compacted in chromosomes. This is cohesing and condensing complexes. I don't want to explain the whole biology here, but there was a very clear thing that this protein called microcephalin was a regulator of condensing by binding on this specific subunit. And nobody was understanding how the function of microcephalin is regulated upon mitosis during the cell cycle. So what we did actually, we used our brain a bit. We got around 50 different proteins that we thought might bind. We ran them, and the one that was binding to this GAPG2 subdomain was this protein M18PP1. And not only it was binding exactly at the same spot as microcephalin was binding. So that was the predicted model. You see microcephalin actually is here, cannot look well, and M18BP1 was there. So we thought that was a true hit. Again, Becky went to the lab, she confirmed it. This is that was done in the native locus for that gene, so it is not an artifact, etc., etc. You see the binding motif is identical between the old and the new protein. We would never be able to find that without this AlphaFill screening. Yes, we did not screen against 20,000 proteins, we screened against the 20 or 30 that we knew from a half-failed genetic screen were there, but still we could only do it because of AlphaFill. And now this is a model. Another group of Alessandro Borsellini, again Andrea Mosacchio, had found similar things. Again, we combined, and again, we published that last week. And actually, since Friday, it's under review itself. Last but not least, you've all seen these PA matrices for the last 5, I'll talk about the 5 minutes. If they're blue, they tell you it's interaction. If there is red, they tell you, well, I don't know the relative distance between these residues, so that means it's not likely interaction. Be careful here, blue does not mean that two things are close. It means that AlphaFill can guess the relative position very well. That is not the same. So this actually PA matrix corresponds to this structure. This is MAP7, that is disorder, but makes a beautiful helix that binds the tubulin diamond in the context of the microtubule. So initially we thought that this was not very handy, and you don't want to be looking at its structure when you run 300 jobs, and we were running hundreds of jobs to find new interactors for different projects. So we came up with these plots that one end is a symmetry average of these two halves of the matrix. It is not symmetric. I know that you can get away not by averaging, by getting the maximum value of the two. You have this matrix that tells us that this project is predicted really well. And here we map the distances between subunits, and you see like here, like this protein, it tells you that it interacts both with A and with B, but A and B only interact a bit, which is reminiscent of this structure. The interesting thing of doing that is that you can use these matrices to identify interfaces in a very quick way, and even when AlphaFill does stupid things like entangles loops and things like that. And then, you know, we have 3 interfaces between alpha, beta, monomer here and the other one. And then actually something that we like a lot is that once we know interfaces, we can make these mass spec-like plots that can show you that MAP7 has an interface with the alpha monomer of tubulin, right? This is the blue one. And with the beta monomer of tubulin, this is the green one. And of course, alpha and beta tubulin has the different interfaces between them. Now we have a web version. I hope very soon we will be able to click which interface we want to see and so on. So that's a way that when you start looking at complexes between 5, 6, 7 proteins without having the accuracy to measure hydrogen bonds and so on, it can be used as a discovery tool, but also the interfaces can be used as a scoring method to find out how true an interaction is. Last but not least, remember, I promised I'd get back to it. Yes, AlphaFill frees out, and if you copy-paste a bit, you get a model. So, DNA, iron, modified based on the DNA. It's amazing. The iron is there. The base that I modified, I only made it from cytosine, methylcytosine. This is wrong because the protein recognizes thymidine. It flipped it out. It freaking flipped it out. I think that's amazing. So the blue domain here should be binding glycosylated thymidine, and there is no glycosylation in the available modifications, so the substrate is wrong. So, but let's look at it. Beautifully flipped out, right? The iron, the active site histidines. Going here, this glutamate is enough to kill the glucose binding part of it. And what was totally amazing, this base, from the base next to the glutamate, what was the distance between these basic 13 base pairs? We published a paper back in 2017 that from whole genome sequencing on single molecules actually, we knew that these 13 base pairs is the glucose and the thymidine hydroxylation that happens by the other domain. And this is actually the way to replicate epigenetic information. Is this perfect? No, it's not. Why? Because you see here, it's the pink string that's recognized, and here is the pink strand, not string, strand that's recognized. This is wrong. It should not be this base, but this base that is recognized. So still, no matter how amazing this model is, it does not explain the biology, it does not explain the biochemistry. Why? Because there should be some bending or winding or moving. There should be something, so we still need an experiment. And I I wanted to finish more or less with that message. I wanted also to say, like, good luck of finding this with experiment. These are 2 microtubule proteins we had, but you will see a few hundreds of them in the last talk of the day, so I will say nothing about the 2. And 5 seconds late, so I will thank everyone. Renzier came up with the AI for finding the metals and the nucleotides. Daniel came up with the algorithm that I showed at the end about the interface scoring and the interface visualization. Ida was the driving force behind AlphaFill validation. Martin is our programmer that programmed in record time all the AlphaFill. And Robbie is not only Mr. PDBU, as you know him, but indispensable in all these projects and supervising the students and Martin. You see that's a few people on the dry land, it's true. We went to the beach, and that was not random. The 3 students that work on the wet lab came swimming. The 3 with the computer stayed in the living room. All true, there was no selection. The protein facility, Patrick was here, Alex Albino, and lots of other people. Many thanks. These are the 3 students that work in the wet lab on projects I really did not talk about. But it's really amazing how well they interact and how each other fits with ideas. So I can go to meetings in beautiful places. Instruct, many thanks for all the support. To all the other people, also thanks for the money. We don't get money from Instruct. INEX Discovery, I will not talk about it. I hope to see some of you in Rome for the final meeting. And mostly I hope that the European Commission, in its infinite wisdom, funds the next program, which is called AI Next. And with that, I want to leave you.